Ladies and gentlemen, please welcome Ari VanderWalde to the stage.
I was pretty close. So a couple of pieces of good news. Number one, for those of you who have stuck around, this is going to be the best talk. Another piece of good news is that a secret to these conferences is that none of the speakers know what any of the other speakers are going to say. So by the end, there's like a lot of stuff that's going to be redundant, and I have no need to actually read every single one of my slides, so I'll get to go a little bit faster, and we're the last one of the day. I was looking at my bio for this conference, which I highly recommend against for any of you who ever need to read your bio. It's like listening to yourself on the answering machine. It's just wrong in all possible ways. And the reason that I'm even addressing it is because the reason that I'm here and the reason that I'm going to be giving this talk is not because of any of the random things that are in my bio, but because of my experience working on a molecular tumor board at a large sort of hybrid community academic cancer center for the last 6 years. So while I'm a director of research and while I have FACP after my name and while I work for various different places, the most relevant thing really is that. And you can read the disclosures if you want to. So what I want to talk a little bit about is how we actually developed a molecular tumor board, where it actually came from, where the idea came from. In about 2011, we started realizing that patients needed to have a little bit more guidance as to what they were going to be treated with in cancer. And I speak as a medical oncologist who was before the immunotherapy revolution, right in the middle of the targeted therapy revolution, where giving people cytotoxic chemotherapy just wasn't the answer. All of you have heard all of this before, so that's the amount of time that I have to spend on this slide, and I'm not going to spend any time on this slide either. It needs to be multi-omic. It's not enough just to do hotspot mutations for EGFR or BRAF or KRAS. You need to be able to do every mutation that's possibly relevant, and these are some of the three that are most relevant. The only reason I'm spending any time on it is because I actually wrote a paper about it a little bit. And a lot of these markers that we're finding for immunotherapy, for example, in this case, don't really fully overlap. So you have to look at a lot of different types of tests with a lot of different types of technology to be able to get guidance as to where you're going to go. Everybody knows that there are lots of different omics. I don't even know how to spell omic. It just doesn't, it makes it seem like, oh my God or something. I'm not exactly sure what omic stands for, but we know that there's a lot of different types of technology that are used in the clinic. And none of these are really all that new until you start getting down to the bottom. As early as 2013, I don't know if there's a pointer on here or not. Oh yeah, there is. As early as 2013, we were doing all of this stuff. All the way down to here, we were able to start doing. Next-gen sequencing really hit the mainstream with the FDA approvals of the Memorial Sloan Kettering Genome Panel on Tumors, as well as the Foundation Medicine NGS panel a few years ago. But these were approved by Medicare. Many of these were approved by Medicare right around the same time, and they were given relatively inexpensively in the clinic for years prior to that. And now we're at the point where whole exome sequencing and whole genome sequencing, as you've heard, as well as proteomics and RNA sequencing, whole transcriptome sequencing are becoming routine and are part of a lot of these tests from companies that are otherwise approved for Medicare and others. How do we decide what we're going to be sending in the clinic? So tissue has always been the gold standard. And for a lot of us, we don't really know any other way because everything else has been a little bit, pie in the sky. But over the last few years, we really started knowing that liquid biopsy is something that we can start making treatment decisions based on. I'm not sure when the scales tipped for that. I would say probably around 2017 in the clinic, we started really using liquid biopsy, usually with DNA analysis to be able to look at it, and now it's really kind of taking off. So in 2019, what does a clinic need for comprehensive or complete metabolic / genomic / proteomic testing? We need to have NGS for about 30 to 50 genes. 5 years ago, I would have told you 5. Now it's about 30 to 50 that are actually actionable for our physicians. We need to know the variant frequency. It actually matters. There are a number of genes, for example, that if you only have 50% of the variants expressing, it means something very different than if you have 100% expressing. It also helps determine whether or not there's a likelihood of a germline mutation together with the tissue mutation. You need IHC, immunohistochemistry, for specific diseases. Obviously, breast cancer needs to have ER and PR status. In addition, PD-L1 is an IHC disease. I can't delve into the problems with PD-L1 testing in the clinic and by every single company, and it's a little bit like democracy, which is the best form of government, except for all the other ones that have been tried. PD-L1 is democracy is the worst form of government, except for all the other ones that have been tried. PD-L1 is the worst immune biomarker, except for all the other ones that have been tried. So that was a really long joke, not necessary, sorry. And then we have to do FISH occasionally, or CISH, either one is fine. We need to be able to get MSI and DMMR status on pretty much every tumor now. That's one of the tumor agnostic tests. We should probably look at total mutational burden. A previous speaker talked a little bit about how we don't really know whether or not TMB is truly a guide. In some malignancies it might be, but it's a pretty good marker to see what the depth and the breadth of coverage is. You need at least 1 megabase of coverage to be able to even estimate total mutational burden. And so if you can get a readout of total mutational burden, it's probably a sign that you have a pretty robust panel. There needs to be annotation for whatever test there is. The annotation is never good enough, but there needs to at least be an attempt by the company doing it to be able to tell you what the results mean. And nowadays, you need to be able to follow these patients longitudinally, which means even if you start with tissue, you need to be able to start doing blood or urine or saliva or sweat. I don't even know what they're going to be able to do, but something that doesn't involve sticking a needle into a vital organ. So what we did in 2014 is we basically integrated molecular testing on tissue to almost all of our metastatic pathways in cancer. And what we did is we implemented it at the time of first diagnosis of metastatic cancer. Not because that's the time that necessarily you're always going to treat patients, but it's the time that you're always going to biopsy patients. So the best practice in the community oncology is that when patients develop metastatic disease, you biopsy the metastatic site to confirm that there's metastatic disease. You're getting tissue anyway then. It's not an additional procedure for patients. That's the best way to be able to get tissue. We basically included about 75% of new diagnoses that at that time had potential findings that could be actionable that we wouldn't have known. And these are all a bunch of alphabet soup that mean breast cancer, colon cancer, lung cancer, melanoma, ovarian cancer, brain cancer, pancreas cancer. And this is basically what our results have been in the last 5 years. So we had about 100 or so molecular tumor boards through 2018. We met every 2 weeks. And when I say we, it means a multidisciplinary group of oncologists in my practice. The practice that I work for sees about 8,000 new cancer diagnoses a year, about 18,000 patients a year. We have a multidisciplinary practice, including medical oncologists, surgical oncologists, radiation oncologists, radiologists, pathologists, and we all kind of converged in this meeting, and it was representatives from each group. We evaluated, using a high throughput mechanism, about 4,500 patients in those 4.5 years, which is a lot. All of those patients had metastatic disease, and the reason that I say it's a lot is because our throughput model was that one physician had to look at the reports that were generated for every one of the patients and decide based on those reports whether or not further discussion was needed in a full tumor board. And the reason I really said it was a lot was because you can probably guess who that physician was who had to go through all 4,438 reports, and his last name rhymes with VanderWalde. So the number of patients that we ended up having passing through for full discussion was about 830, and we generated more than one recommendation for those patients on average, with about 1,000 recommendations made over those 5 years. The recommendations broadly fell. We went into this not knowing what we were going to get. The recommendations we found fell into about 6 categories, really 4 categories, because number 5 and number 6 were relatively uncommon. Either recommended a clinical trial available in our center or in another center, standard therapy, off-label therapy, germline test, and we also, I did not mention, had 100% attendance of genetic counselors, which we have at our center at this meeting as well. And then for rare cases, we would recommend subspecialty pathology review at a disease-specific tumor board, or we would provide advice for cancer that otherwise we didn't know where the primary was, and based on the genomic markers, we could actually give a hint as to where the primary site was. And this is what our recommendations really kind of broke down to. And you can see that, standard therapy, which is 311 of them, that doesn't mean that we shouldn't have discussed the case. It means that there was something actionable in the findings, that we actually had something that was targetable that you could use standard therapy to do. And that's actually quite high. We also had 260 recommendations for clinical trials, based on, most of them were molecularly driven clinical trials, and it actually drove our clinic's choice of clinical trials that we put in our clinic's portfolio because we were able to drive patients and physicians toward these clinical trials. We only recommended off-label therapy 84 times. So if you think about the fact that we looked at 4,500 patients and we only recommended off-label therapy 84 times, it tells you that the fear of these molecular tumor boards, the fear of broad panel testing from payers' perspective, that you're going to end up doing all these tests and you're ultimately going to end up having to give expensive therapy for patients for whom it's not indicated, is actually unfounded. Most of the time, even for the 4,000 patients that we didn't make recommendations for, obviously there was no recommendation for off-label therapy, only less than 5% of patients were recommended off-label therapy. We also then analyzed to see whether or not actually anybody listened to our suggestions. So the way that we put the suggestions out was we put them in the medical record and we emailed the physicians individually and as a group with all of the recommendations that we had. And you can see where the physicians what the adherence to the recommendations were. Clinical trial is always a problem in every center that you'll be in, and only about 60% of clinical trial recommendations were even acted upon. 78% of standard therapy recommendations were acted upon, and that extra 22% is mostly because the patient's clinical picture did not quite fit with what the assumption was going into the molecular tumor board. In Molecular Tumor Board, we reviewed the patient's chart very briefly in real time, but very often our recommendations for standard therapy didn't take into account performance status or the patient's prior history, and so that's why standard therapy sometimes was not done. Off-label therapy, only 37% of patients ultimately were able to get that. Assuming that the baseline is that 80% of physicians want to follow the therapy, the difference there is probably that payers didn't pay for it most of those times. Importantly, germline testing was only followed 36% of the time, and a lot of that has to do with patient concern about having germline testing, and I'll get to that in a second. So clinical trials, can tumor boards serve as a driver toward clinical trials? We made 262 recommendations. Of those, about 2/3 of them were clinical trial now. The patient is eligible for a clinical trial right now, enroll the patient on a clinical trial. About 1/3 of them, about 100 of those recommendations, were for a couple of lines of therapy later. If the patient showed a MSI high tumor, but they hadn't yet gotten first-line therapy, pembrolizumab is approved in a tumor-agnostic way in patients who are in second line. And so we would recommend that you should go on to the clinical trial that we had before it was approved in the second line for that type of trial. And interestingly, 35 patients, which is 13% of the recommendations, enrolled on a clinical trial. That's actually a high rate for clinical trials. In a community oncology practice, the baseline level of a good oncology practice is that 5% of patients go on to clinical trials. So being able to get 35 patients that were enrolled actively onto a molecularly driven clinical trial is actually a win. And you can see a little bit how they broke down. The patients who did not get, so the doctors follow the recommendations a large percentage of the time, but a lot of patients just weren't eligible. So if you notice the discrepancy between the two, that's the reason. Standard therapy, we talked about that already. Off-label therapy is what it is, as I mentioned. And germline testing, what's really interesting about germline testing is we recommended in 143 patients, only 52 patients had that recommendation followed, as evidenced by patients having an appointment with a genetic counselor. Usually the reason that we recommended germline testing was because the patient had a somatic mutation in BRCA or a somatic mutation in a somatic MSI high mutation. We wanted to evaluate them either for BRCA-associated breast and ovarian risk, or we wanted to associate them with, see if the patient had Lynch syndrome or something like that. And what's really interesting is that of the patients who were tested, 35% of them actually had a germline positive finding, which is relevant for the patient and also for the patient's family. And if you just do some basic math, and this is not scientifically or statistically rigorous, that means that if we would have actually tested all of the patients that we recommended for germline testing, we would have had 33 positive patients that we never ended up finding out whether or not they were germline tested or not. So this is a plug for making sure that in cancer in specific, that somatic and germline are more closely integrated. And it is outside of the scope of this talk, as well as outside the scope of the time, to discuss why that might be and why germline testing is underperformed in oncology. So what's next? So one of the things we started integrating in the last couple of years is, of course, liquid biopsies. This is I'm not going to tell you whose panel this is, but this is a breast cancer case that you can actually see over time, the level of the ESR1 mutation, the BRCA2 mutation, all change over time, and you can actually see how they do based on therapy. And we need to do more. And one of the things about these tumor boards is they're not static. You can add things to them, you can subtract things to them as they become relevant. And so hopefully in the next 5 years, after the next 5 years, I'll be able to come back to you and report how we've been able to integrate proteomics, and we've been able to integrate all sorts of different types of peripheral testing to be able to ensure that in the clinic we can put all of these amazing technologies that most of you have been listening about all day into practice and in a more meaningful way for patients. Thank you.